really wanted to focus and devote my career on natural resources, because when I finished my studies, I went back to the copper mine that I used to live. It was getting buried slowly under the earth because of the growing of the copper mine. To see the landscape, the topography of the view that I used to have when I was younger change over time, thanks to the machinery, the constant machinery of the mining. That made me reflect in a very strong way, and I wanted to keep working on that. And that was my first impression of, like, very strong impression of what I wanted to dig in and research. Since then, I've been devoted to first copper, of course, landscape copper, and then when I came to Europe, I wanted to make a parallel with the coal mining. In the economical sense, historical sense, scientific sense, the properties of the minerals, the property of what is energy, what does it mean to provide energy. There's a lot of geopolitics involved, and since I'm away from my home birth, how do I see Chile, my country, where I was born? Because it's a very special landscape. And so then from coal, I moved to saltpeter, which was also naturally extracted from the Atacama Desert. And it's a salt. So then I started to work hands-on with the salt, creating crystals and mixing them with copper, and discovering these new scientific physicalities of these elements. So then that led me naturally to work with scientists since 2014. Yeah, and then since then I've been collaborating with them, meeting different scientists and getting a lot of inspiration from them through conversations. I already wanted to work with bones because I was reading a book by Manuel de Landa, and he put it this kind of like a metaphor, saying that the architecture of the formation of societies and the cities are like the skeleton of a social body. So this sense of calcification in the geological strata, the bony structure as the architecture of the society. So then I wanted to work with both, because for me it was a natural direction, working with minerals, working with the minerals of the body, of a living being. And then I started to gather bones of salmon, and I wanted to crystallize them. And in those 3 months, 2017, all the ideas that I've been developing for the last 6-7 years came to me. So those ideas are the bone, the bonding, the poetry, and the ethos. The ethos, and this is how the collaboration with Karin started. I met Karin at that time in Bergen. One of the things she provided me, besides a lot of information in our conversations, was this role play she developed for her students, and it's an ethical role play. So yeah, this is one of our collaborations with Karin, I would say the strongest.
My name is Karen Pittman. I'm Canadian. I was almost born a foreigner. My brother was born a foreigner. And the one thing that's been constant in our lives is the attachment to the natural world rather than the different countries and political systems and social systems that we've experienced. Well, originally I was interested in ocean biology, and I ended up working in a department of biological oceanography. And I found out, there's an awful lot of biology we don't know. And then I helped to show the first films from the deep sea hydrothermal vents in 1979 in our lab. And there they were showing things that were large animals that were never in contact with light. There was high productivity, and I realized everything we'd been taught about the origins of life is incomplete, and in some ways incorrect. So I thought to myself, how many ways has life decided to be able to come up, survive, and interact with the rest of life? So one of my specialties now is mucosal immunity, that slimy film between fish and corals and everything else in the ocean, which is also pretty well the same thing that's in our guts. So I've been looking at how things interact. One of the things that we've done, and I'm quite pleased about this, we developed mucosal mapping, which is now trademarked, called VeriBar, Verification of Barriers, and gave rise to a small company called QuantiDoc, Quantification and Documentation. And this is about your health. And we're going from analog health to analog histology to digital health. That it's change your perspective. It'll open up the world in a very nice way. Right. Well, remember now, I've had a 50-year-long career. I was interested in aquaculture because that's a real person to animal to environment interaction. So we spent 5 years or something like that mapping out how the small animal is responding to the environment as it's developing. Clearly, I like an audience. You can see that. But I have one in classes when I teach. But that doesn't mean that the communication is getting across. And the way people understand stories is so diverse, I knew that the constraints of a scientific process of communication was in fact self-defeating, because you're writing something in a very constrained and formalistic manner. And so I started working with artists, and I'm looking at how many different ways the artistic way of putting a story out there helps to enhance the scientific way. And then, when we got a project, 1 hour scenic opera called Fish to Mars, and we used a lot of the marine sciences that Bergen is good at, and Bergen's also good at one more thing, and that's metal music. That was part of the working with the artists, and then on the other side, which leads to here. So I've been working with Michel Letelier, who's the artist partner here now. We've been testing a couple of ideas, among them about slime, because the mucosal epithelia, which we don't have on the outside, but fish and all the other animals do, that is a communication layer. It really is. And so possibly that for her is interesting because it's also invisible, but it's quite powerful. There's a lot of science that never, ever reaches the public, and it is quite fundamental. The idea of chemosynthesis rather than photosynthesis. You don't need light, and you can still get nutrition from chemosynthesis, and things like that. And that the aquatic animals wear their immune system on the outside. Wearing your immune system on the outside. Yeah, especially if we remember we're not finished evolving. When you're starting to look at the lifetime of a planet over a long period, then you start to get really humble. You get humble like you do when you're lying on the front lawn looking up at the night sky in summer. That's how humble you get. And so you're trying to figure out how to bring the story across that we're not that important.
My name is Zheng Bo. I live in Hong Kong in a village on Lantau Island. I make artworks in collaboration with plants and, of course, also other humans. I was actually going to study physics, and then when I went to the US, I first studied computer science and gradually moved on to art. So I'm quite happy now that I'm collaborating with scientists, because it's been a full circle. I studied science and then turned to art, and then now also integrating both. I work with plants. This has been the main focus of my practice for the past 10 years. Before that, I was mainly working with communities to think about equality in our social situations. But since 10 years ago, I realized that there are actually many other beings on this planet that I have not got to know well. They are as important as we are, perhaps even more important. So since 2013, I made this pretty dramatic switch to move from socially engaged art, working with other humans, to working with plants. I'm interested in relationships. So first human relationships, and now relationships in nature, in particular with plants. I think that's my understanding of ecology. That's exactly what the ecologists study. They study relationships in different habitats. So there's a lot of common concern. So I learn a lot from research by scientists. But I actually, as an artist, I often are interested in issues beyond what we already understand or even imagine. Because, for example, the ferns in Taiwan, they are very important in local ecology. But because of colonial history in the 20th century, ferns were not present in cultural representations. Artists didn't look at ferns, they didn't paint ferns, they didn't do anything with them in a significant way. They are very important to the local ecology, but also to the local cultural history. So I decided to work with them not only from a biological, but also from a cultural, historical perspective. I was in Berlin in 2020 as the artist in residence at the Gropiusbau. I know that Berlin has great universities, and Germany in particular has great ecologists. So the Sharing Foundation in Berlin helped me to link up with a number of scientists, including Professor Matthias Rillig, who teaches at the Free University and runs the Institute of Biology there. I went to visit him. His lab is in the Botanical Garden. And I also read his research articles. What he does is just fascinating, right? He studies mycorrhizal fungi, relationship between tree roots and fungi. And now he's also thinking about global change. He's thinking about multiple factors of global change, which are highly sophisticated understandings of nature. So I think we clicked, and we just started to have conversations. And I decided to do a film on the political life of plants in a beech forest outside of Berlin. It's a beautiful forest. And Professor Rillig went with me, and then we recorded long conversations as we walk. So I very much hope to continue this relationship, to continue the dialogue, help me to grow these projects and to grow my understanding of the planet.
We work with soil biodiversity in general, but we really do focus on fungi in my lab. And fungi, I think, are probably the single craziest thing out there, because fungi exist as a collection of lines. So they are really so amazingly different from you and I, because when they are in one place and they go to another place, they are still in the place where they came from, and the two places are connected via a line. So these are organisms that exist as networks. And there's a couple of crazy consequences of that. And one of them is that you experience your environment in a whole range of different ways. Is a single cell. And that is one basically manifestation of it, what it means to be a fungus. But the other manifestation is you are that network. And so you need to process information and you need to integrate everything across this entire network. And there is, I think, really nothing like that. And it makes them, fabulously interesting to study. So they're interesting in their own right, of course, but they are also functionally very important in various ways. They are some of the most important symbionts of plants, for example, mycorrhizae, which are some of the things we study in my lab. But also they are pathogens, so they can cause disease in organisms, including humans, but also plants and animals. And they are, of course, decomposers. So without fungi, the nutrient cycles on Earth would come to a grinding halt because nothing would really recycle anything. So they are basically living recyclers as well. They are all different lifestyles that fungi come in. Symbiotic, pathogenic, and then as recyclers. Yeah, we work on a bunch of different types of fungi. Part of the lab works on mycorrhizal fungi, which are fungi that are symbiotic with the vast majority of land plants. Actually, this is a symbiosis that's about 500 million years old and was already probably in the roots of it was already associating with the first plants on land that didn't really have real roots yet. This is known from fossils that are several hundred million years old. So these fungi are much older than the fungi on your pizza, for example. Those are some fungi we work with. They still look the same nowadays as they looked a couple 100 million years ago. And then we work with saprobic fungi a lot. Those are fungi that are decomposer fungi. We don't work too much with pathogens, but we have them always in our repertoire. And when we do molecular ecology, we always also capture the pathogenic fungi. In terms of topics, what we are mostly interested in as a lab right now, we work on really a wide range of topics because the lab is quite large. But a lot of people work on the multiple concurrent joint influences of factors of global change. So there is not only one thing happening at a time, but there is always many factors at the same time that impinge on an ecosystem or any organism in that system or a community. But this is really a challenge to study. You can't use the normal experimental approaches to study this, so it required some new innovations in experimental design. This is what a lot of the lab works on right now. So basically we're trying to understand that entire complex of human impacts on ecosystems, communities, and organisms.
My name is Riikka Tauriainen. I'm a visual artist. I grew up in North Finland in a family of scientists, natural scientists. We have some physicists and some also microbiologists in the family. And this, of course, is already one of the connections because I thought that would be something I would study as well. But as it came out, I became a visual artist. Since my studies, I use a lot of theory as inspiration. It's something that I'm interested in reflecting also in my art. And with time, I came also to work closer with either theorists or scientists. I started working 2018 with the topic of hydrocarbons, which comes from Astrida Neimanis, who wrote the book Bodies of Water. This was in a time where I was working in Italy, in Genoa, and I filmed a lot of sea creatures, octopuses, seahorses, jellyfishes, and I was thinking of the aesthetics a lot, but also our relation to the water, also the feminist aspect of it, also what does it mean in a post-humanist way. So what is a humanism, that the more than human is involved in it. These theories opened up my thinking, also to think as the watery world as a queer world, and this was a very important inspiration. I had the opportunity to go on some research trips to look in the microscope and see the whole microcosmos of the plankton, which of course opened up a totally different relation to these microbic creatures, which are surrounded by us all the time, for example, when we go in to swim or we drink the waters. So we are actually in a very close relationship with these microcosmoses, but we are not so aware of it. The second thing that I also did, I did audio recordings underwater with hydrophones. And also this was a totally new experience and not the one I expected. With Maaike Vogt, who is an environmental scientist from the ETH, we are planning a cooperation, a collective work. It's something that I can do is tell stories. I can make an imaginary trip, which is maybe not just imaginary, also based on the science that Maaike Vogt is doing. Together we can make this transformation, this translation into the art and make it accessible. I think empathy and awareness, responsibility are extremely important things. I'm sure this is also the intention of an environmental scientist.
I'm a marine ecosystem modeler and senior researcher at the ETH Zurich. So that I ended up studying marine ecosystems was mostly by chance. When I finished school, I was mainly passionate about nature itself and I wanted to become an astronomer. So I studied physics. And after I had finished my studies in something altogether different than astronomy in the end, and ended up in a climate modeling group to work on marine biogeochemistry and climate modeling. And I soon realized when I was sent in my very first year of PhD to a field experiment that looking at galaxies, which I loved from a very early age on, was pretty much similar to looking at marine ecosystems through a microscope. And that the small and the tiny is just as beautiful as the large scale. And I understood also how little was known about these ecosystems. And first, all I wanted to do is to model them using numerical models, but I soon understood that there was virtually no data out there to describe these organisms in our models. And then I really embarked on a journey to gather data on these tiny organisms, on who lives where and why, and what they do in terms of marine biogeochemistry. And 10 years later, I now use numerical and statistical models to understand the composition, structure and function of these systems. We're in the United Nations Ocean Decade, so there's a lot of attention on ocean ecosystems at the moment. And yes, there are multiple thousands of researchers in the field being interested in marine ecosystems, mostly those from countries that actually have a coast. And it's an international community that works together quite intensively and very collaboratively. I try to understand who is where and why in marine planktonic ecosystems and how this would affect biogeochemical cycling in the global ocean or all the ecosystem services. The ocean ecosystem is still an unknown. I think we know only a few % of the ocean per se because we are land-dwelling animals. So the ocean is vast and very remote, and in particular it's a 3D environment, so we have depth to consider. So it's still a white spot on our map, despite the fact that we now have satellites in space to monitor all the world we still know very little about the ocean. However, with novel imaging technologies, where we send cameras down into the ocean to take picture of the microscopic life, and also the novel technologies to sample DNA and analyze the composition of these systems by analyzing the composition of the DNA, the amount of information we have on marine ecosystem has been growing exponentially in the past decade. So in 2013, we brought together a collection of data points on observation on who is where and in which amount, where we brought together 1.5 million of observation. And this year, 1 decade later, we've redone this experiment and we have more than 21 million observations that we collected. And that excludes all the measurements from metagenomic or imaging methods. So I would say it's multiple orders of magnitude more. So plankton, they form the base of the food web. So they're the tiny little organisms, some plants, some bacteria, some animals that provide food for all the other higher organisms in marine ecosystem, for instance, fish. And these are the kind of services we get from the ocean, for instance, in terms of food provision.
After school, I studied philosophy first. I realized I was very invested into philosophy, and I still am. For instance, I wrote a book on love, and in this novel, I think about the characters. You know, they investigate: is it possible to redistribute sensual love, just as we redistribute material goods? Could we do it the same way? Give love to those people who are in need. And while finishing the novel, I realized it would be really interesting to try this out. So the context of the art world, also of performance and theater, then allowed me to host trainings. And then at some point, this might lead again into my writing. In the process of trying to make things more real or appear more real, I need help. One example is a filmmaker, Alexa Karolinsky. And trying to explore this idea of the redistribution of love, we shot several films in which we brought a group of people together to explore this task, this challenge, giving love to people in need. The world of fiction lost a lot of its power to get people, to drag people really into your scenarios and make it a bit more real. And funnily enough, often the moment they become a bit more real, people actually see people like in the videos of the Army of Love interacting. I met Alex Jordan first time, I think 5 years ago at a conference, and he was presenting some of his experiments. In particular, the one on the mirror test that he had done with small fishes, and that they were actually able to pass the mirror test. And I was stunned. It was spectacular. I think it was, for me, it was the most important experiment that had been done in the field of biology since many years. Having like fish, like small fish, that like humans usually would overlook, that they would pass this test that was supposed to be like the test for showing that there's like an advanced intellectual mind, an advanced subjectivity self-awareness in an animal was amazing. You know, the mirror test usually is perceived by animals that are able to touch their forehead. You know, they have to touch some other part of their body because there's like a red dot. And I realized, you know, you could sense also from all the other experiments he had been doing that he really knows fish. He grew up with fish. So the approach of Alex and me is to figure out if there's ways that to make humans more empathetic with fish. For this we try to develop a scenario that to create a kind of basic interaction with fish where you as a human kind of take the position of another fish.
Come, yeah, stay with me. Oh. Hi, my name's Alex Jordan. I'm a research group leader at the Max Planck Institute of Animal Behavior, where I head up the Behavioral Evolution Lab, a lab focused in and under the water. One of the primary goals of our research program is to situate ourselves as scientists in the sensory and cognitive and conscious experience of the animals that we study as much as we can. This wonderful word in English, Umwelt, a German loan word, meaning the perceived sensory environment of an animal. And so to do that, we first have to take measurements of the animals, not in a lab, but in the world in which they live, expressing the behaviors that they've evolved to express. And then, once we have a really nice measurement of those behaviors, we can then start to ask questions about what those behaviors are and what they mean. One of the ways we can ask that question is by leveraging some of the advances that we're experiencing in machine learning and artificial intelligence. Now, those advances have to be considered carefully because they can be very scary, and rightfully so. Many of the approaches we use to track and analyze the behavior of the fish and the animals in the environments that we study them in are directly derived from human-based AIs, like facial recognition technology. And some of those, of course, can be used for good, but some of them can be used in a bad way for surveillance and tracking and monitoring human behavior. So one of the ways we use some of these approaches is to track the movement and interactions of fish and break them down into components that we think might be important in their communication, effectively in their language. And then we have a great deal of fun, actually, with what you might consider fish video games, where we build these avatars of the animals and we populate those avatars with behaviors that maybe our neural networks have learned, maybe our machine learning approaches or our deep learning have informed us might be important components to the language of these animals. And we allow those avatars to show those behaviors. And then we can take that experience and give it to a real fish. And we can ask a fish, what do you think this is? One of the most amazing things that we've found is that when we set up these relationships between a real wild fish in Lake Tanganyika and our avatars, we get genuine social interactions. What might start out as an aggressive relationship where the real fish doesn't like this avatar can mature, where they might come to a peaceful truce, and maybe, we're not sure, might even eventually come to be what we might understand as friends. Our goal is to, rather than have animals as our study subjects, have them as our study collaborators to tell us what they are doing and how they perceive the world.
I'm Sister Tolas. My background is chemistry, linguistics, mathematics, and art. And my topic is smell. I call myself a professional in-betweener. My topic is reality, real, and wherever there is air, there is life, and there is smell. So this is what I do since 25 years, and I'm sitting in my archive, in my laboratory, my chemistry lab in Berlin, and this is the epicenter of what I do. So in total, I have up to 20,000 molecules. That's chemical compound, that's smell structures that I've been recording and been collecting over the years. And what you see in the back are part of that. But these molecules are placed in various parts of the lab, depending on the condition they require. The sense of smell is also tricky because there is no language. So it's in human a sense that activates subconsciously. It's not activating the conscious mind. And I also tend to think or know for sure, without emotional reaction, there is no action in this world. What we learn in the context of joy and play and emotion stay with us forever. And what the senses and the sense of smell accomplish is exactly this. So we need to add some joy and playfulness and some, sensory information here, sensory ecology. It's like learning from others, learning from other species. How do they communicate beyond semantic and semiotic? And this is exactly what bacteria and, biology and, all kind of disciplines are looking into. And what we also will do with our project is looking into chemical communication. What does bacteria try to tell us, by being around these days? Are they happy? Are they sad? Or they have other issues that we maybe should understand? And the only way to try to understand it is by active wanting to understand. In other words, scaling down so that we can scale up again. I think the biggest issue is here confidence. How can we bring confidence back to everyone is by really starting to have a discourse around all these things, the micro, macro level of life. It's not just about saying or looking the right way, it's really, you are skilled in smelling. You can smell, this and this information in the air. That's quite something, and give homage to that kind of behavior, appreciation of diversity, in a different way. Otherwise, I'm not sure how we're going to make it. And also with the climate, what are we talking about? How can we understand a tree? How can we understand a bacteria? How can we understand any other animal, living species? When we just continue the way we do and have done so far. So using science and using art to put those issues out there, I think is essential. The level, especially things that we cannot see, that we all know that there is something there that's important. And, viruses are not visible, at least unless you have a microscope. But, we all know they were in the air, that we all were breathing, and the air is a shared endeavor. So we better to take that fact a little bit serious. And I think scientists are ready to collaborate, it's just, we have to find the right purposes, And if for nothing else, also just to find out about this other way of working and understanding the question we all are asking in a different way. So Christina Kapaki is an incredible scientist / artist / human being, who I happen to collaborate with already in the past, in 2010, when she was still at Harvard Medical School, and we got acquainted. We got the opportunity to already then look into potential collaboration, and this collaboration have continued over the years. And what is interesting with Christina, she looked into, she's a biologist, she is working in the biotech, she's working into the sequencing DNAs, and I'm working with the analog world. I'm working with the real, the interface on the body, and she worked with interface of computers and machines. And it's a very interesting conversation we had from the very early day on.
As a scientist, was sort of getting smaller and smaller in my focus, right? Like looking at enzymes and DNA and things at the molecular level, but thinking about problems that are really at this global level. And I found that at that time, the scientists I was working with were sort of like, don't worry about that politics stuff. Don't worry about the people stuff. Like, really worry about the enzyme stuff only. And like, your experiments are the only thing that matters. But artists that I was meeting and talking to, and who were these amazing thinkers asking questions about what it meant to design living things, what it meant to be exploring these new kinds of technologies, they were asking the questions that I wanted to ask too. And so I ended up spending a lot of time working with those artists and thinking with them and asking questions with them, and eventually got connected with Sissel for this project called Synthetic Aesthetics, which was about synthetic biology and art, and using those techniques together and those kinds of bringing the fields together as equal partners to explore some of those questions about social issues and human issues and like things at that more global scale. And so you'll learn more about the Cheese Project.
We're planning to do a project again about identity, about biology, about bacteria and the microflora that grow on your skin, and the benefit of having the microflora on your skin, beyond the notion of it smells good or bad, So looking into the bacteria on the skin, a continuation of what we did in 2010 also with making cheese of human skin bacteria, and for the purpose of changing people's behavior towards bacteria as something negative and dystopian. And the next step of that project is what I intend to do in Switzerland, also because, or we tend to do in Switzerland, also because, the whole notion of Switzerland is like this clean and not perfect, even the cheese is different in Switzerland. So we want to disturb that a little bit. So we hope to find collaborators, be it people from living in Switzerland and also artisans working with cheese. So it's a collaborative project. And if in the end of the day, I don't know if you make sake or cheese or beer, we will see. But definitely we will like to do some excavation into and beyond the surface of various topics in Switzerland.